Okay, so a little bit about introduction. My name is Hartmut Stecher, originally from Austria, so that's the unusual name. I just got a new name today from a Starbucks cup when they write on it. So it's very interesting. I collect those names. It's very exciting. It was a new one today, Stanford one. It was Hartmia. I have the cup right there. Who wants to see it? Most commonly, it's hardwood or hard nut. So, but anyway, I spent the last 25 years of my professional career in cancer research, and I started out in academia. I worked for about 5 years at the University of Washington together with the Fred Hutch Cancer Research Center, where I did basic laboratory research, postdoctoral fellowships in signal transduction and immunotherapy, and then I worked in a clinical research center. And I left academia because I found out when you work in academia, the main focus, and it's good, the main focus is the publication, right? Then I switched to industry, and I worked in the biotechnology industry, multiple of companies working in cancer research, first as a bench researcher for immunotherapy. And it was the first immunotherapy that was brought to market was a cancer vaccine called Provenge. And I worked for this company. It doesn't really work that well. It works in some patients well, but we now understand why it works better. It was 17 years ago that I worked there. And then I switched much more to medical and clinical research, and I did with a lot of oncology throughout the country and also in Europe and Canada, lots of research protocols where we did investigator-sponsored trials in a variety of tumor types. And 5 years ago, I decided when you work in the pharmaceutical sector, the focus is the drug. And I really had pleasure working with cancer patients. And when you work in cancer research, you get phone calls every once in a while from family members, in my own family, and from friends, and you help them out to find a solution. And I found this very fulfilling, so I decided to use my knowledge and the network I built to help one patient at a time to help navigate the treatment options. So you can take advantage of that. I'm happy to work with many people, with all people. I have worked with about 15 ocular melanoma patients over the last 5 years, and even in those 5 years, things changed quite dramatically. So I saw it evolving, the progresses we made. It's slow, but at least it's going in the right direction. People ask me, how do you work? I say, when you have legal problems, you hire a lawyer. When you have cancer, hire me. So I help to really find treatment options and to help you sequence the treatment options, especially if clinical trials get involved. There's inclusion criteria and exclusion criteria. Sometimes you prematurely enroll in one trial, you exclude yourself from another trial. So there's a lot of things. It becomes very complex. You will get inundated from family members and friends, and they mean it all well, and they send you tons of ideas. At the end, you are just completely overwhelmed what's going on. So I use my scientific knowledge to help translate that you can understand and make a well-informed decision. So, with that being said, let's go into detail what I want to do today. I used some slides I presented last year as well to give you an overview of what the key players are in the immunotherapy arena. A little immunotherapy 101 refresher. What are the key players? Then what is an antigen? What is an HLA type? Who has been confronted with, oh, I need to do my HLA type test? Is there anyone in the room who have heard about the GP100 Immunocore trial, Immunocore GP100? Yeah, there's a couple of people. And all of those are very important clinical trials, but what does it all mean? And I try to give you a refresher. And please interrupt me because I'm happy to, if something is still too complex, happy to explain. And there's plenty of time afterwards and tomorrow as well. And then what are those checkpoint inhibitors? You heard about IPI and Nivo, and how does this work and how does it fit in here? And then I will explain the type of T-cell therapies, and T-cell therapies are among the most promising therapies in ocular melanoma. They have approved status in many tumor types, not in ocular melanoma. It's in clinical research, but I think we are onto something here, and I hope that this field rapidly evolves. And Dr. Patel afterwards will also show what is offered at MD Anderson and other cancer centers in the country. And then I give a general overview of what those T-cell trials are and which one would apply to ocular melanoma. In fact, uveal melanoma, because most of those T-cell trials are for uveal melanoma. So the key players in the immune system are cells, immune cells, and they're all part of the white blood cells. You still remember red blood cells and white blood cells. Within the white blood cells, we find the antigen-presenting cell. They look like dendrites. That's why they're called dendritic cells. It's one key player in the antigen-presenting cells. And I will explain what an antigen is. And then T cells. There's different T cells. Some are very naive. They don't have really functions yet until they are programmed to become a mature T cell. And the mature T cell is the soldier cell in the immune system. And they go after the cancer cell. And here's the cancer cell. And many cancer cells together form the tumor. So those are the key cell players in the immune system. There's also key molecules in the immune system. And if you think I overwhelm you now by presenting 1, 2, 3, 4, 5 additional molecules to already 6 different cell types, there's thousands of them, and there's not enough time to go over all those molecules. So those are the key players, and it's a very simplified version. And those are the main players I want you to pay attention to for treatment purposes and treatment planning. Now, how do those molecules and those cell types all fit together? The antigen and the HLA, and I will explain what this is, they sit on the cancer cell, and the checkpoints and the T cell receptor sit on the T cell. So those are important players in the immune system. Now, how does it work that we go from a cancer cell on the very left upper corner to a destroyed cancer cell? And that's the hallmark of the immune system. That's the hallmark how the immune cascade works. And this is how it looks in a raw, and I think I have another one that shows it in a little animation here. So here's the cancer cell, and on the cancer cell is an antigen, and the antigen is what makes the cancer cell unique. An antigen is a molecule that makes the cancer cell unique, and there's many different antigens. And the immune system uses the fact that there is an antigen. Actually, there's many antigens on the surface, but I focus on one. I focus on the Halloween-colored orange antigen. Like my tie, right? They come in all different shapes and forms, and the antigen is the one that is recognized by a dendritic cell. It's one of the antigen-presenting cells. So they find the antigen, they take up the antigen like an octopus with the dendrites, and they digest the antigen. And in the next step, the dendritic cell becomes a mature dendritic cell, and it has special cell types to present the antigen to a naive T cell. That's why it's called antigen-presenting cell. It's like someone reaches out with a plate, and on the plate is a piece of cake. So they present the antigen to a naive T cell. This triggers a lot of chemical and biochemical reactions, and at the end, the naive T cell becomes a very mature T cell and has a very unique molecule on the surface. It's called the T cell receptor. And this T cell receptor is specifically designed to only bind to the one antigen that the dendritic cell presented to it. So you see the very complex cascade, and it's still very, very simplified. But the goal is actually that the mature T cell grows a molecule called the receptor. I call it antenna sometimes, right? And it can reach out now to the cancer cell that has this molecule, the antigen on the surface. And when it does this with a lot of other biochemical reactions and a lot of other molecules in place, it destroys the cancer cell. So the T cell produces a lot of toxic substances that destroys the cancer cell. And this is in fact going on in every body. So we do now understand that cancer is also a disease of the immune system, because as we are standing here, as I'm standing here, there is a very high likelihood that I have cancer cells in me, but the immune system is swirling around and does exactly those sort of reactions and eliminates the cancer cell on the spot. If our body loses the ability to do that, and there's a variety of reasons why this can happen, this so-called immune surveillance stops working and cancer can arise. Any questions so far? Okay, now the tumor can produce a lot of granite blocks in this very important immune cascade. And the granite blocks can be detrimental and nothing works any longer. So those can be substances that can be the so-called tumor environment. And then we have at least half a dozen of steps that the tumor can put in the way of the immune system to stop, and it stops working. And that's where immunotherapy comes in, where we develop different therapies. At the very beginning, for example, we have epigenetic therapy. You might have heard about valproic acid, you might have heard about vorinostat. This falls in the area of epigenetic therapy. It's poorly understood. We have mostly preclinical research data, but it has something to do with some cancers don't even have an antigen or an appropriate antigen. And epigenetic therapy is believed to be in this arena to help develop more antigens on the cancer surface. Then cancer vaccines are more in the area upstream in the immune cascade, where we make the use of dendritic cells to take up the antigen and create an immune system at the very beginning. And then you all heard about the checkpoint inhibitors and T-cell therapies. They're at the very bottom of the immune system. They focus on actually therapies that evolve just T cells and forget about the top. And those are currently in this arena the most promising immunotherapies. And the reason why, it's simply speaking, why T cell therapies might be more promising than cancer vaccine or at the very beginning epigenetic, because all those hurdles we have to overcome in the immune system, we can shortcut. We can shortcut if we focus on the T cells. And we have one of the most promising therapies, T-cell therapies, just recently approved CAR T-cell therapies, and I will explain what this is for a specific leukemia, just got approved by the FDA, and it now has a 90% cure rate for a specific case of leukemia that was incurable just 5 years ago. Now, for the purpose of today's talk, I've focused just on the very bottom of the immune cascade, the T-cell therapies, and what can we do to make these very promising therapy options more prominent in metastatic ocular melanoma. So let me explain what an antigen is. A key player in the immune system is that we have a molecule that is a substance that induces an immune response. For example, an immune response is to recruit a T cell, to make it interesting for a T cell to go after the tumor cell. So that's called an antigen. There is a very complex word called immunogenicity. That's the ability of an antigen to recruit a T cell. So the higher immunogenic a tumor is, the more T cells go after the tumor because it triggers a strong immune response. And that's very important for the T cell response. Now, cancer cell can have their own antigens, or cancer cells can have new antigens. We also call them Neo-Antigens, and those antigens are caused by mutations. A mutation in the cancer cell. In the lifetime of us, there's lots of mutation happening in the body, and mutations make the cell look strange. And a strange cell has the ability to be recognized as what it is, strange, and that's the meaning of the immune system. When we are torpedoed by pollen or any pathogens like bacteria or viruses, and cells that are infected with a virus, they look strange. They look similarly strange as a cancer cell because of Neo-Antigens, and that can trigger a very strong immune response. Now, the problem that we have with Uveal melanoma is that there are not a lot of Neo-Antigens. That's one of the reasons why we believe that immunotherapies and certain immunotherapies don't work well. Compared to skin melanoma or cutaneous melanoma, we have a lot of Neo-Antigen. In fact, skin melanoma is the most immunogenic tumor that we know because it is exposed and originates in cells that are exposed all the time to sun. Sun with their UV exposure has a huge, profound impact to create a lot of mutations. And then the cell, the skin melanoma cell, looks like a Christmas tree. It lights up, it really lights itself up, and it can be found easily by the immune system and by the T cells to trigger the whole cascade that I showed. Uveal melanoma cells do not have a lot of Neo-Antigens. They have some antigens, and that's what we try to use to develop those T-cell therapies and immunotherapies, but they look different. And that's one of the reasons why in many cases the classic immunotherapies like the Ipi-Nivo combinations and other immunotherapies that work in skin melanomas don't work as well in uveal melanoma. Now, important is also one more molecule that I want to mention. The reason why is my experience. A lot of patients call me and says, hey, I had to do some testing. I had to do some HLA testing for a clinical trial. And what is this? HLA is another key player in the immune system. It's basically genetically determined. We inherit all the HLA molecules. Antigen, it's also a substance. Remember, an antigen is just a substance that sits on the surface of a cancer cell that also sits on the surface of almost every human cell. And to make matter a little bit more complicated, the HLA has a function. The antigen molecule, remember the Halloween-colored molecule that we try to develop an immune response against, is basically held by an HLA molecule, and it forms this complex here. The HLA molecule sits on the cancer cell, and the antigen is presented in the complex of antigen plus HLA to the mature T cell. The T cell receptor can only bind to the complex of antigen plus HLA. Now here again, the cell types, the key players and the molecules, antigen, T cell receptor, and HLA. And if we put them all together, the HLA and the antigen sit on the surface of the cancer cell, and the T cell receptor sits on the surface of the T cell. And in a functioning immune system, the T cell recognizes this complex, attaches to the cancer cell, exerts a lot of chemical reactions to destroy the cancer cell. Again, antigen is any substance that can induce an immune response. The HLA is basically the complex molecule that holds the antigen on the cancer cell. And the antigen HLA complex is what is recognized by the T cell receptor. It's pretty simple, isn't it? So what took us this long to find this all out? Now it's still active research because it works. Therapies related to this model works in many patients really well, and in some patients it doesn't work that well, and we need to figure out why. But those are the principles that we use to figure out how we can target uveal melanoma metastatic with the immune system. Now let's go to antigens. I listed here a couple of antigens that we believe will play and are already playing some role in the development of an immune response. And those are own antigens. Those are antigens that are specifically found in uveal melanoma. They are independent of any mutation, so they are not Neo-Antigens. Let's go through the list. It's very simple. MART1. So don't pay attention of why they are called MART1. Usually it's who invented or found the antigen. In this case, it's a melanoma antigen recognized by T cell. It's very simple nomenclature, right? So that's what MART1 stands for. There is GP100. GP100 is a glycoprotein. 100 is the size of the protein. It has something to do with it. And we know that it is usually very common in a metastatic ocular melanoma cell. And it plays a role in one of the promising immunotherapies that are out there. It's not necessarily a T-cell therapy, but it uses the molecule of a T-cell therapy. Then there is tyrosinase. That's an enzyme, and it plays a huge role in all melanoma cells, including uveal melanoma cell, because it is involved in the formation of melanin, which gives the melanocyte, gives our skin the tan color. And that's the hallmark in all of those melanoma cells. Some of them can be very black, and some of them can be less black. And there's interesting research here going on how this has something to do with immune responses as well. There is a fairly new antigen, and Dr. Patel will speak about one trial that she has at her center at MD Anderson. It is a carrier protein and also has to do with the melanin in the melanoma cell. And then there are actually two Neo-Antigens that I wanted to list here. We don't have an immunotherapy against those Neo-Antigens yet, but you have heard about the GNAQ and the GNA11 mutation. Now, in class 2 ocular melanoma, those two mutations are a hallmark. Most, if not all, ocular melanoma patients have this specific mutation. Now, again, remember, a mutation means it looks strange. I do know that Dr. Sato and his team at Jefferson University, they're developing a vaccine. So they want to see if we can take advantage of the fact that there's now a different GNAQ molecule because it's mutated, and can we trigger an immune response? I don't know if any studies are open yet, but at least that's in the planning. And then there's a whole list of antigen that we might discover or newly discover to launch an immune system against it. For example, we all heard about the PRAME molecule. Right now it's a prognostic molecule to help identify if PRAME is expressed in class 1 ocular melanoma, it's a more bad sign. If it's not expressed, it's a better sign. I did not add it here on yet because, to my knowledge, there is no immunotherapy that will be developed against the PRAME, but I wouldn't be surprised if the future will also bring some immunotherapies against this molecule. HLA, you remember, the HLA is the one that holds the antigen on the surface. There's many different HLA types. HLA-A has 1,200, or what does it say here, 1,700. HLA-B has 2,300 members. Those are really the numbers, how many of those exist. And HLA-C, and there's other HLA types. So there's thousands of them, right? Among the HLA, there's an HLA-O2 type, and we have 456 of those. And one of the most common one is the HLA-2 subcategory 1 type. Actually, about 30% of all Caucasians in the United States have this genetically predetermined molecule. So there's some immunotherapies that are developed to the majority who has those type, because remember, the T-cell receptor, all those therapies that involve a T-cell receptor only recognize the antigen in context with the HLA. So whenever you are confronted with HLA testing, we have to first see if you express this one molecule, because otherwise the immune system, the therapy wouldn't work, because the T cell could not attach to the complex of antigen. That's why there is testing required. Now, sometimes they say that this is up to 50% or down to 25%. I think more realistically, with different databases that I lately looked at, it's in the vicinity of 30%. In African Americans, it's only 12%. So the likelihood to qualify for a trial is substantially lower that they qualify because you need to test positive wherever a T-cell receptor molecule is involved. Any questions? Am I too fast, too slow? Okay, so, and to make matter worse, there is one more set of molecules I want to talk about, and those are called the checkpoint molecules. I showed to you that the immune system can be very effective in killing a cancer cell when the mature T cell with its T cell receptor can bind the complex of antigen and HLA, and it destroys very effectively the cancer cell. That's the goal. That's what we want to achieve. And we have many, many therapies now in clinical trials that utilize this complex mechanism. Now, there is two molecules. Actually, there's many more than two, but I want to focus on two that can put a little fly into the ointment of the immune system. They are called the checkpoints, and they can shut down the immune cells. So the checkpoints are just other molecules. They are both on the T cell, and there is certain molecules that can bind to those checkpoints, and they're on the cancer cell. And look, I have two errors here, two errors, very strong destruction of the cancer cell. Now, if one of those molecules, don't remember the name, on the cancer cell binds to a molecule called CTLA4, one of those lightning strikes goes away. We have also a molecule called PD-1, and we have a ligand on the cancer cell. If this binds to it, nothing happens any longer, and the cancer cell is not destroyed. So I repeat this, because it makes it easy in animation. You have CTLA4, and if CTLA4 binds to a molecule on the cancer cell that fits right into the binding pocket, only half of the tumors get destroyed. And if another molecule called PD-L1 ligand binds to this PD-1, which is another checkpoint, then nothing happens and the tumor is not destroyed. It's a very simplified model of two molecules, how this works. In fact, there's now over 30 checkpoints, and some of them are in active clinical testing for drugs. Now, what do we do if this happens? This is one very important mechanism how the immune system can be shut down. We put checkpoint inhibitors to the rescue, and two of the most famous ones are ipilimumab, trade name Yervoy, or nivolumab, trade name Opdivo. And in short, they are also called Ipi and Nivo. And I made in this animation the appearance of those molecules. They don't look like this because they are monoclonal antibodies. But for easier explanation, what happens with those molecules, they are very effective. Ipilimumab fits perfectly into the binding pocket of this CTLA4. And now this molecule on the cancer cell can bind to it. And I restore the first lightning strike. And the nivolumab fits perfectly in the binding pocket of the PD-1, and this can bind the PD-L1, and I regain full destructive capability of the T cell. Now I repeat this as well to make it easy. Ipilimumab binds to one of those checkpoint molecules, prevents the binding of the cancer cell to the checkpoint, and I restore part of the immune system. And the nivolumab binds very effectively in the binding pocket of PD-1, and the cancer cell cannot bind there, and I regain full power of the immune system. That's a simplified model. Again, there's many other checkpoints out there, but those checkpoints are also believed to play an important role in our immune system to shut down the T cell, because otherwise, if we would not have those checkpoints, we would have a completely non-regulated T cell that destroys all the time. And in fact, this is happening, and what we believe is part of why autoimmune diseases get established. And autoimmune diseases can be very destructive. For example, lupus, rheumatoid arthritis, ulcerative colitis, Crohn's disease, psoriasis. All those are autoimmune diseases where T cells are unregulated and going after body-owned cells and destroy them. Unfortunately, that's also the reason when we do this reaction that I just showed you with nivolumab and ipilimumab, we see a fairly high number of autoimmune-like side effects. And that's the biggest problem we face with those checkpoint inhibitors. When we apply them, one of them, we have a certain chance. Two together, Ipi and Nivo together, we need to, in more than 50% of the patients, treat the autoimmune-like effects. It can be colitis, it can be pneumonitis, it can be rash in the skin, it can be serious autoimmune-like reactions, and we need to apply high-dose steroids. And some patients are still on steroids to fight those autoimmune diseases. Now, the problem we have in ocular melanoma, since we have less antigens, we have less T cells. And those checkpoint inhibitors alone won't do the job anyway because we don't have any T cells. So what I see very commonly, and unfortunately I've worked with some uveal melanoma patients that are seen in a community-based setting, where there's not as big a differentiation between skin melanoma and ocular melanoma, and they're treated with Ipi-Nivo. As you saw, the likelihood that it works is very low. 5% is the response rate. But the side effects are still up to 60%. So it doesn't really do the patient any good to immediately apply skin melanoma therapies like Ipi-Nivo combined to ocular melanoma. Okay, Lots of stuff to learn, but remember, antigen is important. We need the antigen to recognize the cancer cell. The HLA type is important to hold the antigen in the right manner, and the checkpoints are important to launch an immune response. Now, I want to focus on the T-cell therapies again. Topic of today's discussion: how can this reaction of mature T-cell binds to the cancer cell and destroys the cancer cell be utilized and made into a therapy against metastatic ocular melanoma? And there's, in general, two types of T-cell therapies. One is called tumor infiltrating lymphocytes, or TILs. You might have heard about TILs. Some patients froze their TILs. And if you have any questions what it is, Dr. Patel will explain right after me what this means. And then there is a different type of T-cell therapy that utilizes genetically modified T-cells. Those are not tumor-infiltrating lymphocytes, but those are T-cells that are taken out of the patient. They're genetically modified with very, very interesting technologies, with retroviruses. That was actually one of my fellowships at the University of Washington, where I developed those viruses in a gene therapy laboratory. Because we can modify T-cells so they develop the right T-cell receptor. So we can make so-called transgenic T-cell receptors, or TCR T-cells, or we can make a completely artificial molecule, and those are called chimeric antigen receptors, or CAR T-cell receptors. And those are molecules that are actually not T-cell receptors. Has advantages and disadvantages. One of the advantages, in general, those CAR T-cells, they're independent of the HLA type. So they can bind differently than the TCR does it. Okay, so the tumor-infiltrating lymphocyte make advantage and use the fact that in any tumor, any solid tumor, and here this must represent a colon cancer, but it's the same for liver cancer or liver metastases, we will always find some immune cells that infiltrate the tumor. And that's actually happening. And most of those immune cells in ocular melanoma are dormant. They infiltrate, they somehow find the tumor, but they don't do the job really well in eliminating the tumor, but they're still there. And that's what the tumor-infiltrating lymphocyte clinical trials take advantage of, and I will explain how this works. And then the genetically modified T-cell therapies, the CAR T-cell and the transgenic T-cell receptor, they actually create an artificial T-cell receptor or an artificial molecule which we call a chimeric antigen receptor. So that's a completely different story than the tumor-infiltrating T-cells. So remember, there's two forms of T-cell therapies: tumor-infiltrating lymphocytes and genetically modified T-cells. Now, the way how we get cells for those T-cell therapies is also slightly different. In the case of the tumor-infiltrating lymphocytes, we usually take a part of the tumor. Here it's represented as a breast tumor, but when we do tumor-infiltrating lymphocyte harvest for a metastatic uveal melanoma, we usually take a biopsy or we snip it, surgically removed part of the tumor, and we isolate the T cells. We separate the tumor cells from the T cells. We grow up the T cells in an infusion bag. That's how it's represented here. We give a lot of healthy growth factors to those T cells. We grow them up into billions, and then we give them back to the patient. In the case of the transgenic, the genetically modified T cell therapies, we usually take T cells out of the bloodstream. There's a methodology called leukapheresis. It's about a 2-hour procedure where you have a needle in one vein and another needle in the other vein. In between is a blood filtration machine that separates the red from the white blood cells. And the white blood cells include the T cells, and we can separate those T cells, and then we genetically modify, and here's a piece of DNA where we add the DNA to those T cells, and we grow artificial T cell receptors, or we grow artificial molecules called CAR molecules, chimeric antigen receptor molecules. And then we grow them up with the artificial receptors, and you see that's the main difference here. those artificial receptors are not the same as the tumor infiltrating lymphocyte. The tumor infiltrating lymphocyte are a mixture of many different receptors, any of those T cells that made it to the tumor. In the case of the genetically modified T cells, it's one specific receptor or one specific molecules that recognize only one specific antigen. We grow them up into billions and infuse them back into the patient. One thing that most T-cell therapies now have in common, we need to prepare the body before we infuse the cells, because nature would be too simple if there wouldn't be all sorts of regulatory cells. And there's a lot of cells that regulate the T cells. In fact, they're called regulatory T cells. And we learned that they actually regulate the T cells, do not do their job. So we have to get rid of them before we give the therapeutic T cells, and we do this through chemotherapy. And in many of those T cell therapies, we give now a non-myeloablative chemotherapy regimen. It's usually cyclophosphamide and fludarabine in a 5-day course. There is variations of this theme. And we do this to get rid of the regulatory T cells. So we lower the white blood cell count, which usually is a fairly harsh methodology. We don't use this high of a chemotherapy that it would destroy the bone marrow, but we need to get rid of the white regulatory blood cells so that the T cells can effectively do their job. Now, I did a search of T cell clinical trials for melanoma that might include uveal melanoma. And what you see here, I listed it based on those antigen. Remember, there is hardly any Neo-Antigens. So there's hardly any Neo-Antigens in uveal melanoma because the mutational load, there's not a lot of mutations in uveal melanoma. And most of the T-cell therapies that are currently open and available for melanoma that might, in most cases, include uveal melanoma, utilize the intrinsic antigens. MART1, which is the melanoma-specific antigen. There is 3 trials that you can find on clinicaltrials.gov. 2 of them are actually including uveal melanoma. Dr. Patel will talk about one of them. And there's another trial that uses a transgenic T-cell receptor, specifically for MART, and it's open. It is at UCLA. However, this trial currently does not recruit any patients, so it's unfortunately not available. I emailed Dr. Ribas, who is the principal investigator. He emailed me back yesterday and says, "Oh, we are not recruiting." I said, "Oh, please change it on clinicaltrials.gov." Then the GP100 trial. GP100 is a very common antigen, remember, the orange molecule, right, on ocular melanoma cells. And this GP100 currently doesn't have any T-cell trials. However, it has various IMC GP100 T-cell-like trials. There is a T-cell enhancer molecule, and this molecule is basically a T-cell receptor that recognizes this antigen, the GP100, and it has another part of the molecule fused to it that binds to the T-cell. So it brings the T cell closer to the cancer cell, hopefully with the same effect as I showed. So I decided to include those. It's interesting to see that there is actually no TIL trials or no TCR or CAR T cell trials. And then I'm happy to forward those trials. I have to move on. Those are currently the trials that are available. Dr. Patel will talk in more detail. The CAR T-cell trials are listed here by the number of antigens. So we have quite a lot of antigens for all solid tumors that are currently, and liquid tumors that are currently in clinical trials. And the most common antigen with about 80 trials, close to 80 trials open currently, is for an antigen called CD19. And this CD19 is only found on B cells, and B cells are part of the leukemia and lymphoma. And there's 2 approved CAR T cells right now. They were just recently approved last month and in August, and it was big in the news. Unfortunately, a lot of cancer centers and those companies say we have now the tumor melting T cells. I get calls all the time from all sorts of cancer patients, mostly solid cancer patients. Can this therapy be used for my case? And I said, unfortunately, you don't have a B cell cancer and you don't have the antigen CD19 I don't know if you can read it, because that's what this therapy is specifically approved. You need a tumor with CD19. We do know that ocular melanoma does not have CD19 on its surface. Now what we need to do is we have to check what T-cell therapies are in clinical testing. Are any of those antigens there? Also interestingly, most of those CAR T-cell trials, the genetically modified T-cell trials, are offered in China. And if you look specifically what sort of, I would say almost 2/3 of all open CAR T-cell trials are in China. And most of the rare antigens where only one trial is open in China. Some of those trials are very, very interesting and might be a little dangerous. For example, there is one trial that has PD-L1 as the actual antigen. Remember, PD-L1 is one of those checkpoint molecules on cancer cells. Now, this could be very critical because it can not only eradicate cancer cells. There is one T-cell trial at Jefferson University, a CAR T-cell trial that targets MET, c-MET. We do know that the molecule itself plays an important role in the metastatic potential for ocular melanoma. And there is an idea that we could use this CAR T-cell and enroll ocular melanoma patients. However, MET is also expressed at other cells in the body, and those are critical things because if we launch T-cells into the body that also attack healthy body cells, that can lead to really bad side effects. Now, the way how we have to do is this is the list of antigens that we know we have in ocular melanoma. Unfortunately, none of them is available in the CAR T-cell trials. So CAR T-cell trials, again, are currently only for the liquid tumors for one specific leukemia, acute lymphoplastic leukemia, and one sort of B-cell lymphoma, and those are the currently approved therapies. And this is the last slide, I promise. And this is a very promising end slide. I wanted to show the NCI and Dr. Kamula, who now is at the University of Pittsburgh, published earlier this year. It was published, I think, in June of 2017, the first interim results of one of the first TIL trials for ocular melanoma. And what you see here is a so-called waterfall plot. Everything that goes down is positive, is good. The tumors actually, in this patient number 16, shrunk by 100%. And the response is long-lasting. So there's no tumors anywhere in the body. There was one patient out of those 21 patients, where the tumor completely disappeared. And this was a patient who was on first treatment Ipi, Nivo, and had no response at all. And it was a devastated patient, but this patient responded really well. And there's some other patients. The red one are all those ones that did not work on Ipi, Nivo alone. So the fact this was a trial, it was a TIL trial. Again, a piece of the tumor was taken out. Out of those tumors were a few T cells extracted. They were grown up into billions of cells, and this regimen was given back into the patient. And that was the therapy. And you see that the majority, 2/3, responded. Everything that goes down shows tumor shrinkage. So if the tumor shrank more than 30%, up to 99%, it's called a partial response. If it's 100% shrinkage, it's complete response. Unfortunately, there's also a few patients who did not respond at all. Some of those patients, unfortunately, recurred, and we need to figure out why they recurred. But this is just to show you that we are going in the right direction, and there's potential. There's huge potential, and that's why we have immunotherapy trials to explore this, to make it better, to make it more specific for the antigen, and to combine treatment modalities. We can combine TILs with checkpoint inhibitors to make it even better. Okay, so that was my whirlwind tour through T-cell therapies for ocular melanoma. Huge promise, huge potential, but still a lot of work to do. If you have any questions, I will be sticking around until tomorrow at noon, and please don't shy away to ask me.